This is an English listening video summarizing 10 sushi science. Enjoy 10 interesting facts about listening to English. Carbon monoxide tuna. Did you know that sashimi tuna can also suffer from carbon monoxide poisoning? The vibrant red color in tuna comes from a protein called myoglobin. With time, myoglobin oxidizes and turns into metmyoglobin, giving the meat a brownish, less appealing color. However, when tuna is treated with carbon monoxide gas, the gas reacts with the iron ion in myoglobin to form a stable compound called carboxymyoglobin. This process locks in a bright red color, making the tuna appear fresh for much longer. While this treatment preserves the color, it does nothing more. As tuna decomposes, it produces histamine, a substance that can cause allergy-like food poisoning. Because the carbon monoxide treatment keeps the tuna looking red, consumers might not realize it has spoiled and could eat it, putting them at risk. From the perspective of consumer protection against this masking of spoilage, the treatment of tuna with carbon monoxide is not permitted in Japan. The Science of Red Vinegar. Did you know that the dark color of red vinegar is caused by the same type of reaction as the browning on a steak, whereas common rice vinegar is made by fermenting sake over several months? Red vinegar is produced by aging sake lees, the residue from sake production, for several years. During this long aging process, a Maillard reaction occurs, creating brown pigments called melanoidins. This reaction is what gives the vinegar its characteristic dark color. The multi-year production process of red vinegar is also known to generate a large amount of amino acids. While typical rice vinegar has a total amino acid content of about 100 milligrams per 100 grams, red vinegar contains more than 10 times that amount, at over 1,500 milligrams. This creates a strong umami flavor while softening the sharp stimulus of the acetic acid, resulting in a richer, more mellow taste perfect for sushi. The K-value. When determining the freshness of fish, people often rely on appearance or smell. However, there is a more scientific method known as the K-value. This value offers an objective indicator of freshness by measuring chemical changes that occur in fish muscle after death. Specifically, it calculates the percentage of certain compounds that result from the breakdown of a substance called ATP. Based on this, a K-value below 20% is generally considered suitable for eating raw, as in sashimi. For fish that will be cooked, a value of up to 50% is typically acceptable. The rate at which the K-value increases is heavily influenced by storage temperature. For example, a horse mackerel kept at 5 degrees Celsius would have a K-value of around 10% after 1 day, making it ideal for sashimi. However, after 3 days under the same conditions, its K-value would rise to approximately 40%. At this stage, the fish is no longer recommended for raw consumption and should be heated before eating. This shows how crucial temperature management is in preserving the quality of fish throughout the supply chain. The Ikejime Method. Ikejime is a traditional Japanese fish-killing technique renowned for preserving the quality of the meat. The process involves swiftly inserting a spike into the fish's brain to cause instant death. This is often followed by threading a thin wire through the spinal cord to destroy the nervous system. The primary goal of this procedure is to stop all nerve signals and prevent involuntary muscle movements that would normally occur after death. This careful handling is believed to be essential for maintaining the fish in its freshest possible state for a longer period. The scientific basis for Ikejime's effectiveness lies in its ability to control biochemical processes. The destruction of the nervous system prevents the fish's muscles from consuming their stores of ATP, an energy compound. This conservation of ATP significantly slows the progression of rigor mortis, the stiffening process that occurs after death. By delaying rigor mortis, the method allows for a period of aging where enzymes can naturally produce umami components like inosinate, enhancing the fish's flavor and texture while minimizing spoilage. The Science of Wasabi. Many people are familiar with the sharp, pungent sensation that wasabi delivers, which quickly travels through the nasal passages. This distinct kick is the result of a chemical reaction. When wasabi is grated, its cells are broken down, allowing an enzyme called myrosinase to mix with substances known as glucosinolates. This interaction instantly produces a compound called allyl isothiocyanate, or AITC. Because AITC is highly volatile, it easily vaporizes at room temperature. The gaseous AITC then stimulates a specific pain receptor in the nose called TRPA1, which the brain interprets as that characteristic sharp sensation. However, there is a clear difference between authentic Japanese wasabi, or hon wasabi, and the more common horseradish-based substitutes often served outside of Japan. Hon wasabi contains another important compound called 6-methylsulfonyl hexane isothiocyanate, or 6-MSITC. Unlike the highly volatile AITC, 6-MSITC vaporizes more slowly. This property is responsible for the milder, more aromatic flavor and subtle sweetness that follows the initial sharpness. Horseradish, on the other hand, primarily contains AITC, resulting in a more one-dimensional and intense pungency without the complex aftertaste of genuine wasabi. The origins of sushi. While sushi is globally recognized as a quintessential Japanese dish, its origins can be traced back to Southeast Asia. The earliest form of sushi, known as narazushi, emerged around the Mekong River basin as a method for preserving fish. Fish was placed in cooked rice, and the natural lactic acid fermentation of the rice preserved the fish for months. When it was time to eat, the rice, which had a strong sour taste and odor, was often discarded, and only the preserved fish was consumed. This culinary practice eventually made its way to Japan by the Nara period. The sushi we are familiar with today is a much more recent invention, born in Edo, modern-day Tokyo, during the 19th century. The widespread availability of rice vinegar during the Edo period eliminated the need for the lengthy fermentation process. Coinciding with the rise of a busy street food culture, chefs began to create a new type of sushi that could be prepared and served quickly. This led to the creation of nigiri sushi, a small ball of vinegared rice topped with a slice of fresh seafood from Edo Bay. Chefs also developed sophisticated techniques known as shigoto to enhance the flavors of the fish, such as marinating it in soy sauce or curing it with salt. Umami Synergy. It is well known that kombu, or kelp, adds a savory flavor known as umami to sushi rice. This is due to an amino acid called glutamic acid. However, when this is combined with the inosinic acid found in fish and seafood, a remarkable effect occurs. The perceived umami taste is intensified by up to 8 times. This phenomenon, called Umami Synergy, was quantitatively demonstrated in 1957 by Dr. Akira Kuninaka, who found the maximum effect occurred when the two substances were mixed in a 1-to-1 ratio. The mechanism behind this synergy is linked to specific taste receptors on our tongues. Glutamic acid binds to a receptor, sending a basic umami signal to the brain. Inosinic acid then binds to a different part of the same receptor. This second binding action essentially locks the glutamic acid in place, preventing it from detaching quickly. This extended activation of the receptor dramatically increases the intensity of the umami signal that is sent to the brain, resulting in a much richer and more profound savory flavor. Data-driven sushi. Did you know that at some conveyor belt sushi restaurants, the plates themselves report their own travel distance? At the chain Sushiro, IC tags on plates were used to track freshness, with a famous rule that any sushi traveling 350 meters was automatically discarded. Furthermore, analyzing the data from over 1 billion plates a year, an AI could predict demand for 1 and 15 minutes in the future. This system successfully reduced the chain's food waste rate to just 1%, a figure much lower than its competitors. However, this seemingly revolutionary system has an ironic conclusion. Recently, the chain has shifted away from circulating plates on the main belt, instead having customers order directly via touch panels for immediate delivery. This has made the problem of unsold circulating plates obsolete, bringing waste from that source down to zero. From one perspective, the complex and expensive data system was merely a detour before reaching the simplest and most effective solution: to stop the practice of circulating the sushi altogether. Mussel sea bream. Mussel sea bream is a type of farmed sea bream with increased muscle mass, created using genome editing technology. This fish was developed in 2016 by a research group led by Professor Keitaro Iedo of Kinda University's Aquaculture Research Institute. By deactivating the function of a gene called myostatin, which suppresses muscle growth, they succeeded in increasing the fish's muscle volume by 1.2 to 1.5 times compared to conventional sea bream. This technology was developed with the goal of producing a larger amount of edible fish meat more efficiently. The texture is said to be unchanged from ordinary sea bream, remaining soft and delicious. Unlike genetic modification, no external genes are introduced into the organism. Therefore, the process is described as artificially inducing a mutation that could also occur in nature. While it is expected to help solve food shortages, providing clear information to consumers remains a key challenge. Sushi robots. Did you know that sushi robots, which are a symbol of inexpensive sushi, actually require very advanced technology? The act of shaping sushi rice is a delicate process that is difficult to replicate with a machine. The ideal rice ball is firm on the outside and soft on the inside. It must be solid enough to be held with chopsticks, yet fall apart gently in your mouth. This texture is achieved by incorporating about 40% air into the rice. Simply compressing the rice with a machine would crush the grains and create a hard lump. To solve this difficult problem, manufacturers have developed their own unique approaches. Suzumo Machinery, the company that developed the world's first sushi robot, uses a shaping part made of urethane rubber to reproduce the soft touch of a human palm. It also uses a special comb blade method to portion the rice, which protects the air layers. Meanwhile, Fujiseki focused on the process before shaping. They developed a spiral loosening technology, which uses a spiral-shaped part to gently mix the rice in the hopper. This prevents the rice from hardening under its own weight.